to share with you the results of the last decade of research at Imperial College, where we began investigating the genetic basis of bioactivity and have learned that it may be possible to use this finding to stimulate a second revolution from just replacement of tissues to regenerate tissues. This offers the real possibility of solving the most difficult global challenge, I think, that is facing all of us here today. The challenge of a social revolution that for the first time in human history, we are reaching a time when there will be more people depending upon the industrial support of less than 1/2 of the population. This revolution is a result of the huge increase in survivability that has occurred during the last 100 years due to the result of many changes in lifestyle, immunization, clean water, many factors, but as a result, the numbers of people worldwide in the developed countries that are living past the age of 60 are going to be approaching the numbers of people that are in their productive ages from 20 to 60. This is already happening. And half of the people that are in their productive age now, at least in the UK, work for the government. And so, and that's becoming a trend as well. So the consequence of this revolution of living longer and longer is a sacrifice in the quality of life, often. I mean, most of you that are here have parents that are suffering the consequences of this increasing lifespan, which is the decreasing quality of our connective tissues, particularly the cardiovascular tissues as well. But we will be primarily talking about the 40 years of revolution in at least dealing with the increasing fracture probability that as we get older, and particularly is true of women, bones get weaker because the structure or base of bone, such as this trabecular bone that are in our vertebrae at the end of our long bones, as we age, gets weaker and weaker, and this leads to this type of phenomena of the shear of bones, the long bones, the shear of the femoral head, and this incapacitates hundreds and hundreds of thousands of people every year. The need is enormous, and that need is increasing every year. It is not just a static need. We're talking about millions of implants that are required. We need a new approach to the problem. Well, in the past, in fact, St. Mary's Hospital, which is across Hyde Park from Imperial College, this was how the type of problems were solved. And that was in a previous government, of course. And now, of course, the present is, instead of just cutting off limbs when they hurt or such forth, or cutting out parts of the body, the current procedure used worldwide when needs occur is to replace tissues. And that replacement has become a big business, $20 to $30 billion a year business. 30 to 35 parts of the body are now routinely replaced by some 20 to 30 different types of materials, and it is continuously referred to as sort of, many cases, miracle replacements. Most of you know somebody that has a knee replacement. Our first speaker, Joe Miller, this morning, we had a little chat. He said that he would never have believed when he was growing up as an engineer that he'd be walking around at his age with a titanium ultra high molecular weight polyethylene knee. And he was, when he was at DuPont, he helped develop the ultra high molecular weight polyethylene that he is now wearing. Okay? In fact, you might sort of say a little prayer. You hope you never live long enough to have to use the stuff that you're actually working on inside yourself. And it turns out that this next year I'm probably going to be a candidate for a Bioglass periodontal repair on a 10 millimeter defect in one tooth that they tell me if I don't change my eating habits will get worse. So, well, the standard approach of tissue replacement had been, up until 40 years ago, to use bioinert materials, such as the metallic parts that are shown in this hip replacement, or polymer parts like we are looking at here. So metals or polymers were the norm. But the future is not to use exclusively bioinert materials or even the bioactive materials that we will do a short review on here, but to actually think about using the body's own repair mechanisms to regenerate our own tissues. Now, this would have seemed to be an impossible dream even a few years ago, let alone the 40 years ago. But this is where the future lies. It is to turn on the body's own repair mechanisms that get progressively poorer and poorer as we age, but to use bioactive gene regenerating materials, which we will be looking at examples of in just a few moments, to restore structure, function, and thereby the biomechanical and biochemical properties to regenerate the tissues inside ourselves rather than in replacing them. Well, the approach to this problem began, as I mentioned, 40 years ago in November, when we made this discovery that in the heart of this compositional diagram, which is not a phase equilibrium diagram, you won't find this in the American Ceramic Society phase equilibrium book, it is a kinetics diagram. And I submit that one of the approaches that we should be thinking more of in ceramic educators and in the ceramic research community is to create kinetics diagrams. The center of this kinetics diagram, that oval region, is the bioactive region for class 1 bioactivity, that all of the compositions of glasses made in this compositional range, a soda, calcium, silica, with a constant 6% phosphate, all will bond to bone. And they'll all bond to bone very, very quickly. And we now know from the recent work on the gene expression that the slow dissolution products released from those glasses will activate genes to turn on the repair of bone. When you get outside that boundary, you can see another broader region, which is also bioactive. Compositions in that region will also bond to bone, but they bond slower. They don't bond to soft tissue, and they don't release enough silica and calcium to activate the genetic repair processes to quickly regenerate new bone. The other compositional ranges are bioinert materials, or if they're non-glass forming, they would be simply rapid dissolution. Well, we learned in the first year or two after the discovery, in around 1970 to 71, that this phenomena of bioactivity was related to the surface chemistry of the glasses. The special composition range of glasses, when exposed to body fluids, or even just distilled water, would quickly change from a surface of an amorphous material to a growing calcium phosphate layer, which would then crystallize to form bone mineral on the surface of the glass. A physical-chemical process. A physical-chemical process that precedes any of the biochemical processes. And that is a very important discovery because it sort of mimics what we believe happened in the natural world as the geosphere began eventually, after several billion years, to be populated by life forms. Physical chemistry changes that occurred in the geosphere that led to the biochemical changes. And it's those biochemical changes that we now are learning activate genes. And so we are rediscovering, perhaps, the fundamental phenomena that led to life formation and the consequences of that, which are sentient human beings that are here. We learned also in early work, and in fact, Carlo Pantano, who's here somewhere, I saw in just a moment, wishes he had copyrighted this SEM that he did at Florida at a postdoc, because I show it all the time. It is evidence that the collagen fibrils that are incorporated in the surface of the growing appetite layer become woven into the surface in a structure almost equivalent to what happens at the interface between bones, tendons, and ligaments in the body. And this phenomena happens without the presence of cells, right, Carlo? It's simply done with exposing the surface of the glass to the collagen in a solution. So again, the physical chemical binding was the key. And for years and years, we thought that that was the most important key. We learned that the interface between bone and this monkey segmental bone replacement tested under impact torsional tests, the same sort of torsional load of impact that the Penn State quarterbacks get when they face the Ohio State linebackers. And fracture occurs, in this case, not at the interface between the bone and the implant. And this finding was extended and made into a very important product. Professor Kukubo, standing back there, this is a slide from the pioneering work done by Professor Yamamuro and Professor Kukubo, producing a very hard, strong material, sufficiently strong for vertebral replacement. And this is showing the bone actually growing to their material without the fracture of the material. So we're still thinking about replacement of tissues, the interfaces, in this case a whole year long after an implant of the material, with the bone cells lining the surface, growing on the calcium phosphate and the silica-rich layer. And then it was discovered that soft tissues would also grow these material, discovery made by my late wife that many of you know, June Wilson. And that led to the understanding that the collagen in the body would bind to these active surfaces, whether it was type 1 collagen, like in bone, or type 2 to 4 collagen in other tissues, which gave rise to enough understanding to get ethical permission at the University of Florida to do clinical trials, which were then extended to Guy's Hospital in London, which began a long relationship with research and the clinic in London. And the first FDA-approved products of the use of bioactive glasses for replacing the bones of the middle ear. And then another year later, in mid-80s, replacing teeth that have been extracted before dentures are being placed for the elderly patients. But we're still talking about replacement of tissues. And that was the dominant thinking throughout the community as we shifted from emphasis in bioceramics from inert materials like alumina, zirconia, which are still very important implant products in hips and knees. But we're still thinking about replacement until another discovery was made in the late 1980s by periodontist, University of Florida, Sam Lowe, and June Wilson and myself. They mostly did the work that was possible if you replace powders of the bioactive material in the presence of a bone defect, that it would lead to the formation of new bone around the powders, connecting together, forming bridges. And that connecting of those bridges led to new bone, rapid new bone generation, which was submitted to FDA for approval for use in repair of periodontal defects, gum defects. And this is the surgical slides from Dr. King Smith showing that after just 6 months, a large periodontal defect on the one side has now been completely healed and repaired and has saved the tooth. And in today's social revolution, social economics of the cost of repair and maintenance of the quality of life is a #1 concern. Most countries no longer can afford to maintain the quality of life of the millions and millions of aging population. How can we cut the costs down? And this is one example of cutting those costs down. If that tooth had been lost and had to be replaced by a bioinert titanium implant, that patient or somebody would have had the cost of anywhere from $5,000 to $10,000. Instead, the periodontal regeneration has only cost a few hundreds of dollars. That's the shifting in the thinking that we all need. The oral surgeons soon learned that they could also fill larger defects using the bioactive glass powders, and this is only a few months of a defect that was the result of a surgeon extracting a impacted third molar in a teenage girl, and the bone now is completely regenerated, that area, without requiring the need of a autograft from the iliac crest of the patient, which was the standard procedure at that time. Much more expensive, and then eliminating the pain of a second site surgery to take bone from the patient to fill that site. Orthopedic surgeons soon began to use material, and FDA has now approved orthopedic applications, and hundreds of thousands of cases are using the bioactive materials. And this is an example where there was a large bone defect in the bone surrounding this implant that would have led to failure of the implant, and a cost of $20,000 to $50,000 for the repair if that implant had failed. Instead, the surgeon removed the cyst, put the bioactive glass, now trademarked as Nova Bone, in the site, and that repaired and saved that implant from being lost and avoided revision surgery. So there are now about 20-some products sold in 40-some countries worldwide using the bioactive glass, the same grandfather composition we discovered 40 years ago. The material is being sold under the trademark of Nova Bone. It can be simply used as a powder, or also now as a putty. It is used in orthopedics for various types of trauma repair, for arthroplasty of filling around implants, for general surgery, and particularly important in America, where there's a growing need in our aging population for spinal repair. 500,000 procedures are being done now annually, requiring inner body fusion, and using a synthetic material as an alternative to the patient's own bone, particularly for the elderly patients, for the post-60. God, that's elderly. It includes at least a few of us here in the front row. This need is growing annually by far greater than gross national products and healthcare costs in general. Craniofacial repair, there are whole ranges of applications. I won't go through that whole list where they're used. So U.S. and worldwide, there are a whole host of products, each labeled with their own country's names and titles and such forth. There are also other alternative products that are related, very close relations to the original 45S5 Bioglass products that are being sold. Nobody knows for sure, as far as I've been able to ascertain, but there is I think a gross estimate that somewhere between 5 to 10 million patients have had their quality of life enhanced by the use of this generation of bioactive materials. 12 years ago, though, we began a quest, and that quest is the second objective today in this talk, is to share with you, because it offers the promise of opening the windows for this dealing with the global impact of this aging population worldwide. And that is to try to understand at a fundamental molecular biochemistry level, the mechanism of bone growth called osteogenesis. And we had a hypothesis that it was genetic stimulation by the ionic dissolution products that lead to the growth of the new bone. The first quantified evidence of this is a result of international collaboration between Professor Nishi's surgical team at University of Osaka, Japan,
Japan, and our group at Imperial College at the time, where he developed a very important model of critical size defect in rabbits, where the defect is filled by the powders, and it made it possible to quantify, as a function of time, the kinetics process in the animal of filling and regenerating that bone. And comparative studies were done of all the synthetic materials during Professor Onishi's period of directing this work. And this gives one example of the huge difference between the class 2 bioactive materials, where bone will grow, but only by a process of so-called osteoconduction. This is 1 week of a synthetic hydroxyapatite made in Japan, commercially on the market, used by surgeons. In 1 week, this yellow line, you can see there's very little, if any, bone that is penetrated inside that defect in 1 week's time. And in contrast, the class 1, class A, bioactive 45F5 Bioglass, grandfather material, within 1 week, there are bridges between these particles penetrating at least halfway or more into the defect, creating the new bone. Rapid colonization of bone on the surface of the particles. We now know that that bone is following the concentration gradient of the release of the silicon and calcium at critical levels, the turning on the genes of the progenitor cells that are present at that site in just 1 week's time. After 12 weeks, the whole defect is completely filled and is remodeled. New bone is there. Some of the particles still remain. Most have been resorbed and replaced by the new regenerating bone. It opens up the concept of a completely new approach. And when you look at the kinetic curves of comparison of these different materials, we find that with the use of the class A bioactive class materials, that we have the same amount of bone quickly formed within a couple of weeks as was in the defect before it was trephined out by the surgeon. So we began to think, why does that happen so quickly? How can we control that, and how can we use that information? And particularly, what is the genetic basis of this? So we compared the material science with the biological science, and again, in a kinetic expression of log curves here, and I don't take the time to go through, but the phenomena was that the implant we were seeing was producing a slow release of critical concentrations of the silicon and calcium that were turning on, we hypothesized, the genes to lead to cells in a cascade to proliferate and grow and form the new bone, and that produced the new rapid bone growth. So we began testing, with the direction of Professor Julia Polak, myself, this hypothesis, that there were two things happening within this regenerative environment. Intracellular effects, things that were taking place inside the cells due to the release of the dissolution products that were turning on the growth process. And so we began this investigation with support from U.S. Biomaterials, and we quickly learned a number of very important things. That the cells from the primary bone cells removed from femoral heads of old patients, old being 55 average here, which at that time seemed old, now it seems pretty young. They would grow, quickly form a new bone by 4 steps of the cell cycle, all steps influenced by the bioactive glass. This is a big shift in the thinking. You don't just put the material there and then wait for the body to respond to it. In this case, the material is turning on the cells to shift their cycle. And the shifting of the cell cycle would lead to a complete sequence of events that led to proliferation. And this has now been animated with the help of a graduate student, and it's the first time I've seen it presented publicly. And this is now, in a short time clip, what happens when the glass is put into the body. A progenitor cell gets turned on if it's capable of forming new bone. If it isn't capable, it's turned off and dies and gets out of the way. There's some real socioeconomic implications here, right? Okay.
The new cells, the daughter cells, these two osteoblasts, then if the environment is right and the glass provides the right environment, they can then stimulate both the production of new cells, more cells which are needed, and the differentiation of the cell to grow bone matrix around them that mineralizes, that forms the strong composite, living composite structure of bone. So three things can happen in the presence of a bone cell in a site, regardless of age. Whether it's young people or old people, three things can happen. If the environment isn't right, the cells can die and get out of the way. If the environment is right, they can divide, proliferate, and begin to form new tissue. And then when the environment is really correct, they can mineralize and form new bone. But that is a very complicated sequence of events that is under genetic control, that is present at the time when the egg is fertilized and we all begin become an individual. It was found that this process can occur in culture, in a test tube environment, in the presence of the primary bone cells with the bioglass particles. It can actually form colonies of the bone cells that will mineralize. The gene expression experiments then began to prove that this was under genetic control, and the first experiments were relatively small. Only 1,450 genes were analyzed. Subsequent experiments were done and were published using half of the whole human genome. 30,000 genes were analyzed in the presence of the bioactive gel glasses, the sort that Julian Jones is working on, in the presence of human embryonic stem cells, in fetal cells, as well as old people's cells. In all cases, these results showed the same, that there were families of genes, in fact, 7 families of genes that were activated or upregulated by the presence of the glass. And this upregulation and activation occurs within the first 24 hours of exposure of the cells to the glass. A rapid response, which if we can learn to use this same understanding, we have the potential to regenerate all of the failing tissues of the body, to regenerate the heart, the cardiovascular system, the lungs. There are no solutions to chronic lung diseases now. But the possibility of using this information and this new understanding that it is possible to turn on the body's own regenerative processes give us the potential for solving this global challenge that is due to the social revolution that we inherited from our parents and grandparents. It gives us all the hope of living to 80 to 100 years of age, but with a quality of life that is more than just sitting around in rest homes. As I said, it's been established now for embryonic stem cells in the presence of all sorts of the bioactive stimuli. And so I want to conclude this celebratory talk with an acknowledgement. I started to make a list, and it would have taken the whole 40 minutes to go through the incredible list of students, a number of which, former students which are here in the audience today, and faculty colleagues, University of Florida and Imperial College, and many, 20-some research groups worldwide that I've had the great privilege and honor to work with over my years. Oh, I can finish with to say, hey, it's been fun. Great. Thank you. Thank you.
Thank you very much, Larry. I think there may be time for one question, because we know that we are standing between lunch and the session. So somebody has maybe one question, a very quick one.
Where can they buy it?
Yes.
Oh, yes. I'll repeat the question. It's a very important question because I think I went through it too fast. The critical ions that we now know are responsible for activating the genes are soluble, biologically active silicon ions and calcium ions, and each are required in a certain range of around 15 to 20 parts per million of the silicon and about 60 to 80 parts per million of the calcium. Those two ions are what are involved in activating the nuclear response to the cells that leads to the differentiation. If you're outside that range, too low, class B bioactivity or no bioactivity of all. If it's too much, the material then is overactive, and so it's a critical concentration. Thank you. Of silica, calcium.
Okay, thank you very much, Larry. Let me give you this.
Okay, great.
This small token of appreciation from the Methodist left side.